Hello, my name is Veria Alvarado, and I work with Dr. Herman Scholtov group at Texas A&M in the Department of Plant Pathology. And we are studying the zebra complex disease and mainly the different populations of psyllids. So 2 years ago, we joined the group for this research goals, and we did find out that the psyllid populations, the psyllid is the insect that transmits zebra complex disease, the psyllid populations have different amounts of the Liberobacter. Liberobacter is the causal agent of zebra complex disease. So we start monitoring these colonies. It means that during time we will collect samples of the psyllids and we will check if Liberobacter was present or not, and we find out that there were colonies that have high levels of Liberobacter on them, and there were colonies that have low levels. So we also observed that there was a correlation with the amount of the individuals in the population. So the hot colonies have a reduced number of population, and the clean colonies have a high amount of individuals in the population. So we decided to study what was the microbiota composition of the psyllid. So the Liberobacter is like a part of the flora of the psyllid, like we have in our digestive tract. So we know in our digestive tract we have many different bacteria. So psyllids also can have the same happening on their tract system. And so we start looking into the dynamics. If the Liberobacter increase the population, is anyone else affected? Is there another microbe in that gut that is less abundant? So we start exploring the microbiota composition dynamics, and we start looking at the Wolbachia. Wolbachia is one bacteria that is known to change these rates of reproduction. So far we haven't been able to consistently find a correlation with the Wolbachia and the reproduction, but not all the colonies, we obtain similar results for all the colonies, but a recent colony we obtained from Dalhart is showing us that Wolbachia may be involved in these proliferation behaviors. So there is another possibility that we are also exploring, is a phage. Phage is a virus that resides within a bacteria. And when the phage becomes lysogenic, it means that it will destroy the bacteria. So we are gonna be looking if the phage is present in the Liberobacter, and if this phage is being induced by any stress that will cause this decrease in the population of Liberobacter. So these are the main things that we are doing. Besides, we are exploring proteomic analysis with 2D gels. This is looking at the profile, at the whole profile of proteins of plants that are infected and non-infected. And for that, once you compare, you can determine which proteins has been induced by the disease, and if the genome of this plant is available. So if we know the sequences, we can determine what protein is that we are isolating. So this can give us advantage for finding the mechanism of defense in the plant.